So next up, we have Gabrielle Martineau, and she's going to be doing a session on diving into the reef, corals, microbes, and bleaching. Gabrielle is a 22-year-old marine scientist from Montreal who has done lots of cool things like sailing around the Atlantic Ocean on a 70-meter long sailing vessel. She's done expeditions to study marine ecosystems in the Arctic and in French Polynesia. She recently earned her degree and will pursue her career in marine biology graduate program at the Hawaii Institute of Marine Biology. She's also a member of the World Oceans Day Youth Advisory Council and an intern at the Canadian Space Agency. So without further ado, I'll let her begin her session. Thank you, Gabrielle.
Awesome, thank you. Can everyone hear me?
Yep.
Okay, let me share my screen a second. Okay, can everyone see my PowerPoint right now?
It's good for me.
Okay, awesome. So let's start. So my first question to you guys is, have you ever had the chance to dive on a coral reef before? If so, I would like you to please comment in the chat your favorite dive site to date. Do I have some divers in my audience? If not, that's fine also. Okay. Oh, I have someone. Yes, Pinnacles in the British Virgin Islands. Awesome. I have never been there. Seems like a beautiful place. I'll wait a second to see if I have other divers. Okay, I don't think so. So, if you've never had the chance to dive on a coral reef before, I'll tell you what it's like. It feels like you're surrounded by a colorful and vibrant world with a consortium of alien-like organisms of all shapes, colors, and size, ranging from primitive to evolved, and from microscopic to colossal forms of life. Those organisms are swimming around you like they're on a highway of a city that is bustling with life. One of the most fascinating shows on our planet. So coral reefs are not only dynamic and beautiful, but they are also of high importance for both our human health and oceans, our human population and ocean health. Sorry. 1 second. Okay. So even though they cover as little as 0.2% of our ocean surfaces, coral reefs are one of our planet's most productive ecosystem and act as major biodiversity hotspots, sheltering about 25% of the world's marine biodiversity. Sorry. So this is mainly due to their complex architecture and to the many nooks and crannies in which many fish, worms, and other invertebrates shelter, find food, and rear their youngs. Besides, reef habitats are also where most of the seafood we eat begin their life. In a sense, coral are the cradle of marine life. As a general rule, the more complex the structure is, the more biodiversity it can harbor. So to me, one of the most fascinating part about that biodiversity is that most of it is actually undetectable to the human eye, at least on the surface. As an example, a couple of years ago, I was working on a reef hunting for fish, and I thought I was staring at a coral, while suddenly I saw a big moray eel just springing out of nowhere of a cavity inside a reef. So this is just an example of how surprisingly diverse our reefs can be. So here are some little videos of the diversity that we can see on a reef. For instance, these here are some of my favorite worms. They're Christmas tree worms, and their way to cope with predators is to actually go into their hole as soon as you poke them. So they're everywhere on the reefs in the Indo-Pacific. I love them. So coral reefs are also important for our climate. So they are often compared to the tropical rainforest as they are an important source of oxygen production. They are the lungs of our blue planet. Corals are also equally important for humans' population worldwide, as they deliver an array of ecosystem goods and services upon which millions of people rely on for subsistence and economic growth. So coral reefs provide a source of protein and income for over 5 million people worldwide. In addition, for a lot of people, reef ecosystems are also tightly linked to the culture and to the way of life. Plus, many new drugs that are created to help humans come from corals. As an example, bryostatin that comes from the coral rhizomes is used to fight against cancer. And according to many scientists, we are only at the start of the discovery journey of new drugs in corals, prompted by the novel chemistry found in coral reef ecosystems. So coral reefs also act as wave buffers, so they can reduce wave energy by up to 94 to 95%. They can hinder island erosion and maintain our coastlines intact. Concurrently, they consist in a line of defense against storms, hurricanes, and other natural disasters. Basically, without reefs, the islands and the coast would be crumbling. So as of now, we have talked about the reef ecosystem, and I will introduce you to the coral organisms themselves. So what are corals? Corals are the foundation species of the reef ecosystem and are responsible for erecting its complex tridimensional structure. So to picture this, we could easily compare coral reefs to forests. In this case, the trees would be the coral animals and the forest would be the reef. So corals are made up of 1,000 of individual structures called the polyps. So here's a picture of a coral polyp that's stretching its tentacles. A thousand of the structure called the polyps form together the colonies that we can see here. And after years of growth, the colonies build a reef. So as pictured in this animation, younger islands form what we call a fringing reef. So in this case, the reef is directly attached to the shore of the island. As islands get older, they can form what we call a barrier reef. So in this case, the reef is separated by the island by a lagoon. In some cases, reefs can form atolls. So in addition, some islands are entirely made of corals. So there exists a high diversity of corals that exhibit a high diversity in structure and shape, that is morphology, which are each associated with distinct habitats. So for instance, the coral genus Acropora is often characterized by an arborescent morphology, as we can see here, as corals that belong to the genus Montipora are often encrusting. And those are only a few examples of the broad diversity that we can find on coral reefs. So the coral health and homeostasis, that is the equilibrium, strongly rely on its composition, described by this association between the cnidarian host animal and its symbionts. So the symbionts are divided between the coral microbiota and the symbiotic algae from the family Symbiodiniaceae. Together, those three components form what we call a coral. So the coral host animal, or the polyp, consists of a circular mouth surrounded by tentacles. There are very, very ancient animals that belong to the phylum Cnidaria. The coral host animal supports its symbionts, that are the microbiota and the symbiotic algae, by providing them with safe, well-illuminated niches, as well as nutrients such as nitrogen and inorganic carbon, in addition to metabolic byproducts. So I will show you an animation that actually pictures the tree of life and that depicts the large evolutionary distance that separates us, Homo sapiens, from the Cnidarian phylum. So to me, the most fascinating part is that corals, cnidarians, are actually really ancient organisms, but they're still incredibly complex. So here are our corals. The coral lives in intimate partnership with a microalgae that resides in its inner tissue, as we can see here. And we call those algae symbionts. More specifically, those microalgae are dinoflagellate of the family Symbiodiniaceae. So the Symbiodiniaceae in turn primarily provide the coral host with oxygen and photosynthetically fixed carbon, that is sugar, in addition to be responsible for the formation of the coral skeleton through the precipitation of calcium carbonate. So in a sense, those symbiotic algae are the food factories of the coral. The family Symbiodiniaceae shows a great diversity in the reef ecosystem and encompasses 7 different genera. So among those, 4 principal genera are found in corals, and those are commonly found in corals, and they are circled in red. So we have the Cladocopium, Brevolum, Turridinium, and Sangodinium genera. So this is actually a phylogenetic tree of the family Symbiodiniaceae. So the coral microbiota consists of a consortium of other microorganisms, bacteria, archaeas, viruses, and other unicellular eukaryotes. The microbiota is namely involved in nutrient cycling, metabolism, and stress tolerance via a broad spectrum of mechanisms. So, okay, I will try something. Let's do a little poll. So I would like everyone who's there to try to vote for which one of these stressors do you think is the primary cause of coral deterioration worldwide. So you can answer, I don't know if this will work, using the reaction emojis here. If it doesn't, I actually don't know if you guys are able to react, but.
Yeah, I don't think so because it's a webinar.
Okay, sadly. Well, we'll skip that part. But anyway, you will get the answer soon. So corals are highly susceptible to various stressors that are imposed on them. So corals' ability to keep up with the rapid pace of environmental change is poor in comparison with other species, hence the crucial need to study and understand those valuable organisms. So first of all, one of the threat imposed on corals is dynamite fishing. So dynamite fishing contributes to eradicating coral reefs. Namely, repetitive and extensive blasting events in the same area are particularly destructive for corals because corals don't have the time to recover between each blasting episode. In addition, introducing chemical contaminants, sewage disposal, heavy metals, pesticides, herbicides into the reef can modify the entire ecological equilibrium of the ecosystem. In fact, coral reefs thrive in oligotrophic waters, that is, waters that are poor in nutrients. So adding organic matter to this system will stimulate the degradation of that organic matter into nutrients by bacteria, just as the same way that we can see in soils, for instance. That increase in nutrients will catalyze primary production and algal growth. That algae can be epiphytic algae and cyanobacteria, for instance. As coral and algae are in competitions for habitat space and light, the rise of algae will shift the equilibrium towards the algae and will be detrimental for corals. So if you're familiar with the process of eutrophication in the lakes, this can be a bit similar. So on top, plastics and microplastics, including lost and abandoned fishing gears, are imminent threat to our coral reefs. So those materials are ingested by even the tiniest organisms and are transmitted to predators of higher level in the food chain to end up in our plate via a process called bioaccumulation. Also, increased rates of sediments can cause particles to fill up the gastrointestinal cavity of the corals, that is the mouth, limiting its capacity to capture prey. Sedimentation can also increase the turbidity of water, which can be problematic to the growth and survival of corals, since it contributes to reducing the amount of light that reaches the polyp, hence limiting their capacity to acquire food through photosynthesis. So I'm going back a bit with plastic pollution. So just to illustrate how bad of an issue plastic pollution is, I was able to notice in myself while I was sailing across the Atlantic. So we were in the middle of the Southern Atlantic Ocean, extremely far from land, and we passed by a floating fridge. So that tells you a lot about how much plastic there is in the ocean, and you can only imagine how much plastic there is near our coast. It is actually astonishing. So another threat to coral is the demographic boost of some predators. This has been documented quite a lot with the crown of thorns starfish that has been held responsible for massive coral reef decline over the past years. The starfish has been preying on corals and ravaging reefs worldwide. So storms are another threat to corals. So in addition to the mechanical destruction of coral reefs, storms, hurricanes, and other natural disasters contribute to the erosion of reefs and to a general input of sediments, which are impeding coral growth and survival. Finally, like any other organisms, coral can suffer from diseases. So viruses and pathogenic invasions are usually spurred by a rise in ocean temperature and an increase in nutrients in the waters. Let's talk a bit about overfishing. So an ecosystem is in equilibrium. Consequently, a small shift in abundance or the removal of or the addition of one of its components can cause large-scale changes. So here I'll provide you with an oversimplified yet self-explanatory example of how trophic ecology works in a coral reef ecosystem. So for instance, let's say we fish a predator of high trophic level. Let's say here we are fishing the snappers and the groupers on a reef. We will observe what we call a trophic cascade via top-down control. So in this case, coralivores fish and macroinsectivorous fish aren't fished on as much anymore compared to the herbivores on the reef. Consequently, the relative proportion of herbivore on the reef will diminish. Nevertheless, herbivore fish control the development of algae on a reef and hence exert a control over the algae-coral competition, shifting the equilibrium towards the algae. In addition, if coralivores don't get preyed on as much, their abundance will increase on the reef, again, shifting the equilibrium towards the algae since they feed on corals. In the same way, if we increase the abundance of microinsectivorous fish, they will prey on urchins, hence again, shifting the equilibrium towards the algae, in this case, towards the crustose coralline algae. Let's talk about climate change. So with the burning of fossil fuel and the increase of atmospheric concentration of CO2, we are observing a rise in ocean temperatures around the globe. Those issues are of great concern at the moment and require a call to action for humans' population and for our oceans. So at present, our ocean act as buffer to trap the heat generated by burning fossil fuels and by emitted greenhouse gas emissions. Our oceans actually trap about 93% of that heat. So the question is, what will we become once the ocean exceeds its buffering capacity? Quite concerning. So as a result of global warming, we are observing an increase in frequency and severity of climatic perturbations, such as marine heat waves and dryness on land. So those marine heat waves are causing our corals to bleach, leading to an increase in susceptibility to opportunistic microorganisms and higher mortality rates in corals. Thermally induced bleaching is the primary cause of coral deterioration worldwide. That was the answer to the poll, by the way. So concurrently, coral bleaching is an imminent threat to coral survival and is attributed to an important reductions in coral cover. So in 1998, our reefs have experienced the first global massive bleaching event on which we lost 16% of our corals. Our coral reefs literally became graveyard. In 2015 and 2016, following the El Nino event, we saw an increase in 4 degrees in ocean temperature. Thus, in 2016, 29% of the corals on the Great Barrier Reef have died in a single year. That was just devastating. More importantly, according to the IPCC, that is the Intergovernmental Panel on Climate Change, they predict a rising in global ocean temperatures between 0.4 to 1.1 degrees by 2025, potentially resulting in catastrophic impacts on our reef ecosystems. So a lot of people are actually not aware of the enormity of these issues. As we lose coral species, we are losing genetic diversity that those species need to adapt to environmental change. Without corals, there will be no more reefs. So let's dive literally into corals' mechanisms of bleaching. One might wonder, what on earth is coral bleaching? So coral bleaching is simply a stress response triggered by the coral by its environmental stressors that are imposed on it. Just like us humans would get a stress response if we get a cold or a fever, for instance. The main causes of coral bleaching worldwide are climate-driven marine heat waves, causing coral to switch from a state of eubiosis, that is the healthy state of a coral, to a state of dysbiosis, that is the degradation of corals. So as ocean temperature rises, we observe the breakdown of the obligate symbiosis between the algae and the host animal, leading the white calcium carbonate skeleton to be visible underneath the host transparent tissues. So hence why the corals are becoming white. As corals lose their ability to photosynthesize, they lose their main source of food, and which causes starvation. Overbleaching, coral reproduction and growth are impaired or completely stop. When the coral died, they get covered with microalgae and become a pile of skeleton. So it is important to note that bleaching doesn't always lead to death. In some cases, coral can recover from bleaching and rebound back to a healthy state. So when you dive on a bleach reef, it's actually one of the saddest things. You're witnessing all the life surrounding you just dying, and it gets me so emotional. The most ironic thing is that meanwhile, some people are just laying on the beach, drinking margaritas and living their best life, and actually ignoring what is going on right next to them. So, however, we are finding some corals that remain unaffected by the condition of stress that cause other corals to bleach. We say that those corals are resilient. In ecology, resilience is the capacity of an ecosystem to respond to a disturbance by resisting damage and recovering quickly. In the case of bleaching, we say that those corals are thermal tolerant. One of the main questions addressed by coral biologists worldwide in the past year has been, why do some corals survive and some perish to bleaching? What distinguishes them from the other ones, but mostly, can we understand the mechanisms underlying coral thermal tolerance? So at present, many studies have explored the mechanism underlying long and short-term thermal adaptation of corals. In some cases, potentially allowing for an acclimatory response of the coral under stress. So in this regard, many hypotheses have been explored by many marine scientists worldwide. Some studies have suggested that a thick tissue layer or mounding morphology of the coral has been associated with recovery from past bleaching events. Other authors have suggested that some species were more resilient than others to bleaching due to their shape-dependent differences in colony mass transfer efficiency. Concurrently, other studies suggested that coral that were able to switch to an heterotrophic feeding strategy during bleaching or recovery, that is, capturing their prey in the water column with their tentacles, have an ecological advantage while bleaching, since, as I have explained, energy reserves are limited while bleaching. So again, to dive further into this topic, I will present a brief overview of my research project on coral symbiotic composition under environmental stress. I have been working on this project over the past year alongside Dr. Caroline Dubé and Professor Nicolas de Rome at the Institut de Biologie Intégrative et des Systèmes here at Laval University in Quebec, Canada. So, in addition to the previous hypotheses mentioned, compelling evidence suggests that coral microbiota contribute to the coral thermal tolerance under heat-induced bleaching, that is the microbiota. So some research also suggests that the coral's bacterial community can confer a buffer capacity by maintaining the coral host's vital functions when exposed to stress. So as part of this process, the corals can adapt via the dynamic restructuring of their microbial community and the flexibility of their symbionts. So some other authors have suggested that the switching and the shuffling of different types of symbiotic algae community can also consist in adaptation mechanisms to bleaching. Hence, the coral symbionts play a key role in the coral's homeostasis, health, and resilience. So, yet the respective contribution from the coral animal host genetic background and its microbial symbionts on coral thermal tolerance during a natural bleaching event remains unexplored. Addressing this contribution is of high importance since microorganisms' shorter generation time can allow corals to genetically respond more swiftly to changing conditions, conferring increased resilience to the corals. Filling those gaps in knowledge is crucial to mitigate corals' decline in the age of a rapidly changing ocean, and given the recent increase in frequency and severity of bleaching events worldwide. As such, in our project, we evaluate the flexibility of symbiont communities across distinct host genotypes and their influence on coral thermal tolerance. So, our study was realized using the model species, the fire coral Millepora platyphila. So on this picture, you can see the 2016 massive bleaching event in Moorea, French Polynesia. Over this massive bleaching event, 61% of the coral colonies have bleached. I'm sorry, my PowerPoint is kind of being weird right now, but anyway, okay. So interestingly, the fire coral colonies, for the most part, remain healthy, while other corals, such as the Postlepora, as circled here, did bleach for the most part. So some recent studies have suggested that the association of the coral with the thermotolerant symbiotic algae of the genus Symbiodinium could explain the fire coral thermotolerance over that bleaching event. Okay, sorry, I was just reading the chat again. Okay, so our study design was based on a previous study conducted in the same area by Dubé and collaborators in 2017, and that study was aiming to understand the clonal structure and dispersal and the sexual and asexual reproduction patterns of the same species of fire coral. So essentially, the authors have sampled more than 3,000 fire coral colonies across different reef habitats that were further georeferenced and mapped with an X and Y coordinates. Following that steps, the authors have actually genotyped that sample using 12 microsatellites in order to identify the clonal colonies. So here, 2 individuals are cloned if they have the same identical genetic background, that is the same genotype. So in fire coral clones, clones are obtained by asexual reproduction via a process called fragmentation. As such, the authors have identified more than 300 clonal lineages in this research. So over the 2019 massive bleaching event this time, the fire corals colonies were exhibiting distinct phenotypes, that is, distinct bleaching status, so either bleached or healthy. So consequently, we went back in the field to sample again the same fire coral colonies that have been mapped and georeferenced by Dubé and collaborators. So we have sampled a total of 195 corals that we have re-genotyped. Among them, we have identified a total of 27 distinct genotypes, and 10 of them had a sufficient amount of samples in order to process, in order to conduct our studies. So that led us to a total of 86 samples with 10 genotypes. So we after that, we proceeded to DNA and RNA extraction. So DNA has enabled us to examine the taxonomic diversity of the symbiont communities and gives us the answer to the question, who is there in the symbiont community? While RNA enables us to examine the functional diversity of the coral symbionts and answers the question, who plays an important role in the community? Following those steps, we use metabarcoding techniques followed by bioinformatic analysis in order to characterize the symbionts communities using the 16S rRNA, ribosomal RNA, and ITS2 gene amplicon sequencings on both DNA and RNA for bacterial and Symbiodiniaceae communities. I won't go into further details with those methodologies, as not all of you are from a biology background, but I will present you my results. So today I will only present some results that are obtained on the microbiota community, since the rest of the sample for the Symbiodiniaceae are still being processed at the sequencing platform. So here we can see the microbiota community composition associated with the fire coral. So here, actually, we can see the relative proportion. The bar graph represents the mean relative abundances of each bacterial families for each host genotypes and phenotype found on the x-axis. We were able to identify more than 20,000 bacterial taxa. So the most abundant bacterial family belong to the classes of the alpha proteobacteria, the gamma proteobacteria, the spirochaetia, and the bacterioida, which have all been previously associated with coral reefs or corals. So those bacterial taxa play a major role in the cycling of nutrients, such as nitrogen and sulfur, and are essential in nutrients acquisition, which is of high importance for both the coral host and its symbiotic algae, since, as I talked about it before in the presentation, corals evolve in oligotrophic waters that are poor in nutrients. Later on, we have conducted analysis of beta diversity in order to assess the differences in taxonomic structure of the microbial communities for different host genotypes. And those differences are measured by a distance called the Breaker's distance. So we are able to visualize those distance matrix by using NMDS, so the types of graph that I'm showing you here. So they are non-metric multidimensional scaling. And in this type of graph, each point represents here a sample or a colony. And the points are dispersed according to the dissimilarity of their bacterial communities. So on the left, we have the healthy corals, and on the right, we have the bleached corals. And in both cases, we can see strong cluster of the point of the same colors, and the colors here represent the different genotypes. Further analysis statistical analysis were performed in order to test the contribution of host genotype to the bacterial community structuring. And for that, we use PermaNova, so a permutational analysis of variance. For those who know a bit about that, we used the R software in the vegan package. So for both phenotypes, we had significant results. So together, those results suggested a significant contribution of the coral host genetic background to the composition of its bacterial communities. Similarly, we also did beta diversity analysis, this time in order to assess the differences in taxonomic structure of microbial communities for different bleaching status or phenotypes. So here, the different colors represent the different bleaching status. And again, we can see a strong clustering of the points of the same color. And we again have performed a PermaNova that was again significant. So together, those results suggest a significant contribution of the coral bleaching status to the composition of its bacterial communities. After, we have used the R program statistical package index species in order to carry out analysis to assess the specific bacteria taxa and significant association with distinct host genotypes. So to do so, we selected bacterial taxa that were significantly associated with one or several genotypes under a threshold of 0.01. So on the x-axis here, you can see the samples that are grouped per genotype. And each point here actually represents a specific bacterial taxa associated with that given genotype. And the darker a point is, the more abundant the specific taxa is in the given sample. So we have found 1 to 33 bacterial taxa specific to the host genotypes, while no specific bacterial taxa were found for the genotypes 4 and for the genotypes 8. Of course, after the goal was to know, but which were the bacterial families that were corresponding to those specific indicator bacterial taxa. So for that, we have again used a stack bar plots here that reveals a high taxonomic diversity of those specific bacterial taxa in association with distinct genotype, but that represent less than 5% of the bacterial communities in total associated with the fire coral. So again, those specific bacterial families belong to the classes of the alpha proteobacteria, gamma proteobacteria, Spirochetia, and Bacteroida, which have mainly been identified to be vital symbionts for both the coral host animal and for the Symbiodiniaceae, as discussed previously. In the same way, we carried out analysis to assess the specific bacterial taxa in significant association with different phenotypes or bleaching status. So here on the x-axis, you can see the bleached corals on the left and the healthy corals on the right. So we have identified no specific bacterial taxa for healthy corals and 66 specific bacterial taxa in bleached corals. So again, we have seen high taxonomic diversity of those specific bacterial taxa that represented this time less than 3% of the bacterial communities associated with bleached corals. So we have identified the family of the Rhodobacteriaceae in association with the bleach colony. So the Rhodobacteriaceae have actually previously been described to live in organisms and in warmer waters and can thus be linked to thermal tolerance patterns. And their presence and bleach colonies could also suggest their importance in nutrient acquisition in bleach colonies, since, as I explained previously, those colonies are limited in nutrients due to the loss of their symbiotic algae. So interestingly, we have also identified 2 bacterial families that have never been associated with corals before, but that have been associated with anoxic and extremophile environments. So those environments can correspond to the phase of dysbiosis found in bleach corals. Dysbiosis is the deterioration of the coral. And furthermore, the limitation of oxygen in the bleach coral host tissues due to the loss of the symbiotic algae could also explain the presence of those bacteria. So it will be interesting to measure the functional activity of those specific bacterial taxa in association with bleach colonies that are found in low abundance using RNA. So this will be our next step for this project. This will enable us to draw a parallel between the taxonomic and functional importance of the bacterial communities in association with the fire coral over a natural bleaching event. So globally, those preliminary results, if we can call them, have enabled us to detect a significant contribution of the coral host genotype and of its bleaching status on the composition of its bacterial community. And we were also able to identify bacterial symbionts that might have a potential role in the fire coral thermal tolerance, which is also pretty exciting. So for this research, I wanted to thank the entire team of the Rome Laboratory, especially Dr. Caroline Zbé, Sylvie Bouslama, Pierre-Luc Mercier, and Professor Nicolas de Rome for enabling me to work on such a passionate project. Now I'm going to present you another small research project. So massive heat waves do not only affect coral organisms themselves, but they also affect the species that depend on corals. They can depend on coral reef for sheltering, feeding, breeding, or rearing their young, for instance. One of the main effects of coral bleaching is that it reduces the nutritive resources that are otherwise available for tropical reef fish who feed on coral, such as obligate corallivores, that are fish who feed primarily on coral polyps to survive. So among those, the butterfly fishes family, that is Youth-A-Thon family, are common reef fishes in the Indo-Pacific. So the effects of coral mortality and coral bleaching on coral-dependent tropical reef fish species remains poorly understood to date. It is critical to fill this gap in knowledge, since similarly to what I have explained before with the notion of trophic cascade in coral reefs, the decrease of abundance, or in this case, the local extension of corallivores, for instance, generates an entire reorganization of the entire reef fish community. So consequently, alongside professors Jordan Casey and Simon Brendel, we took advantage again of the 2019 massive bleaching event in Moorea in French Polynesia in order to study the impact of coral bleaching on butterfly fish's behavior and diet. So to do so, we have quantified a feeding behavior in obligate corallivore butterfly fishes, so in two species that are Chaetodon reticulatus and Chaetodon ornatissimus, over 2 field seasons in 2019 and in 2020 that occurred respectively during and after a bleaching event, again on the island of Moorea in French Polynesia. So we took video surveys and shadowed a focal fish, recording its feeding behavior using a camera on 3 different zones of a barrier reef system. We have further analyzed the video using the Boris video analysis software, allowing to measure bite rates and assess the bleaching status of each coral colony that has been fed on. In addition, we were able to describe the feeding preferences of the butterfly fishes by describing the benthic composition of the focal colonies that have been fed on. So in brief, we have identified the corals that have been fed on to the genus level. So either they were acroporas, postloporas, montiporas, or porites, or other corals. So I thought it would be interesting for you to see what a day in the field on the island of Moorea looks like. So the first video is actually one of the video I had to analyze, and here that we took on a reef survey. So here we can see butterfly fish, also Chaetodon ornatissimus, feeding on a partially bleached Postlepora colony. So you can see the reef are beautiful on the island of Moorea. They're really, really diverse. I was extremely lucky to go work there. And the second video is actually simply one of the boats, right? One of the many boat rides we did in order to reach our sampling site. So even though it is rainy, you can still see how beautiful the island is. So it is possible that due to bleaching, we observe a shift in the diet of the butterfly fishes, who may prey on different coral species that are less nutritive, but yet less susceptible to bleaching, in order to compensate for the decline of their favorite species, for instance, that are namely branched corals, such as acroporas. So aside, in some cases, as coral bleach, they emit a range of different neon colors. So this is what we can see in the colonies that are circled in white. You can see that they are neon greens. So actually, the pigment comes from a family of proteins that live in association with the coral host and that have particular optical properties. So the production of those pigments is actually regulated by light intensity. And when the coral bleaches, as I have explained before, it loses its algae symbionts. And hence, the host skeleton receives more light, which actually causes the coral to turn neon, since there are more pigments that are produced in that case. So this process is also a mechanism that helps the algae to move back into the coral during the bleaching event. So some evidences also suggested that those pigments might cause the coral outer mucus layer to be high in sugar. Hence, we have hypothesized that butterfly fishes might switch their diet to feed on bleaching coral colonies. So we are currently analyzing the data, and will be able to validate or invalidate those hypotheses soon. So this project was englobed in Professor Jordan Casey's postdoctoral project that focuses on using fish gut content metabolite coding to analyze trophic interaction and reconstruct coral reef food webs on the island of Moorea in French Polynesia. So Despite all the devastating impacts of climate changes on our reefs and on our ocean, there's still a glimmer of hope. On top of documenting the changes happening to best advance knowledge and inform our policymakers, scientists are now tackling the problems to the source and are attempting to accelerate the adaptive rates in corals through what we call assisted evolution. The goal here is to develop a breed of coral that is resilient to bleaching in the lab and to use those supercorals to transfer them in the ocean in order to restore the reefs. So this can be done by creating hybrid coral by interbreeding resilient corals together, for instance, as we can see here. Or we can also use trained coral via an exercise regime by actually subjecting them to high ocean temperatures that it would experience in an ocean heat wave. So here we can see that we are putting corals in higher level of temperatures over time, and some corals survive and some corals die. So the adult corals who survive actually produce coral larvae that are more resistant to changes. However, there are several challenges with assisted evolution, namely the fact that there's a high mortality of coral transportation as when they go from the aquarium to the reef. And there's also the fact that there's a lot of physical perturbation, for example, due to wave energy or cyclone that is present in the ocean and that is absent in tanks. That is problematic for the moment. So how can you help the reef? To me, this is not at all about donating money. This is about dedication, education, and science communication. So I'm a big advocate for science communications, hence why I'm here today. And I believe that by getting more and more people to know and understand the baseline of an issue and its significance on a global scale, people have the tools to now stand together and take action. So I encourage all of you, even my pals who are living in landlocked cities, to talk about it. To talk about the state of our corals and how it shapes our ocean health in human populations, to share publication on social media, to get people to know what's going on. It takes 2 seconds and it's free. I also encourage you to be your own advocate and to convince the community that this is important. In addition, if you have the chance, go see it by yourself. Get in touch with nature and go dive on a reef. I once had a professor who told me that there's nothing better than going in a field, going into the ocean to best understand and paint a global portrait of what's going on. And I think this is very true. So in addition, we of course need to have our carbon emission under control in order to prevent other massive ocean heat wave, for instance. So even though this is not intuitive, we need to associate global warming with coral reefs decline every day. So every time you use your car and burn fossil fuel, every time you buy a piece of clothing, every time you eat at McDonald's, every time you use a plastic straw, this is contributing to the problem. So small actions such as every time you get a chance, avoid plastic straws, bring your own utensils, buy your Coke in a glass bottle instead of a plastic bottle, encourage sustainable fashion and encourage local fashion brands. Try to carpool with your friend instead of going to a place with 2 separate cars, for instance. Those are simple actions that make a big difference. So there's no magic wand, there's no special potion here. There's nothing that will save us easily like that. The oceans are the lung of our planet and we need to act now. So in addition, you can look up the World Ocean Day website that has plenty of interesting resources, namely the 30 by 30 petition. The 30 by 30 initiative is has actually been created in order to support 30% of our oceans by 2030. So you can go sign that if you want. And overall, keep smiling because there's still hope. So thank you everyone for your attention, and please don't hesitate to contact me if you have any reactions, comments, or even any concerns, you can find me under this handle on Instagram, on Facebook, or via email. And I'm happy to answer any of your questions now if you have some. Since we have a couple minutes left. Oh, someone just said Mauritius. That's awesome. I've never been there. Oh, someone in the chat. So informative and in-depth. Thank you. Thank you, Laura. So I will be there a couple more minutes if ever you guys have some questions. Oh, there's something in the chat. Do you have a favorite species of coral? That's a good question. Actually, corals are super hard to identify. As of now, coral scientists actually need to do DNA work in order to identify the coral at the species level, because it is now visually too difficult to do so. But let's say my favorite genus is Acroporas, because I think they're so beautiful. They're the arborescent form of corals, and I also love rain corals because I think they're really funky. But I think that's the diversity of all of them that makes them beautiful. As cheesy as it may sound. And thank you everyone for the great comment. Oh, Q&A, we have something here. So is using dynamite fishing illegal, or is it common practice in some places? So weird. I'm actually not sure at all about that, but I think it's illegal. I'm not sure though. But definitely it's still a common practices in the islands, since it is apparently efficient to get a lot of fish at once. So, but it is a really, really destructive practice. I'm very impressed with your species pronunciation. You must be a French background. Yeah, in fact, my mother tongue is French, so maybe that's why my Latin is good, or quite good, decent. Yeah, so I'm still available for a couple more minutes if I have more questions. And thank you. I want to thank you all for attending. I hope that you have learned something. Another question is, could you list off the chain of events losing one great reef would have on our planet, please? I'm not sure I'm understanding well the question. I don't know. Okay. So what I think the question is, what would the impact of a loss of a great reef would have on our planet? So I'd say the major impact would be on biodiversity, for instance, and all of the other impacts that I mentioned before. So the thing is that, as I said, most of the species in the ocean and the species we eat, for instance, start their life cycle on a reef. So even though some species are not always on the reef, they might go there only to rear their youngs, for instance. So if there's no more reefs, there's actually no more habitats for those species to go mate and to go sometimes feed and sometimes to go rear their youngs. So that will cause like a major that would be major dramatic change for a lot of several species. So some species may be eradicated, for instance, a shift in abundance, and that will influence all the different levels of the food chain. Since, for instance, like I showed in the trophic cascade PowerPoint slide, either the high level predators and piscivorous fish will be affected, and even the smallest organism, just like the algae. And this is also true for the fish we eat that live in the open ocean, since even those fish migrate, they also use coral reefs sometimes to rear their young, for instance. And this is only the impact of biodiversity. There will be tons of impacts, namely with the fact that, or related to the fact that corals actually they act as buffer for waves. So it could be a major threat for our islands that could be submerged in waters, hence For instance, that would be dramatic for humans population, and if there's cities on this coast, that would be devastating, for instance. So those are just some examples of what would happen if we would lose one of the major reefs on our planet. Does that answer the question? Like, there's so many aspects of it. Like, I think I could spend like an hour speaking about those consequences. But one thing that is sure at is that it's not good. It's not good at all. It would be devastating. So yeah. So I have I think 3 more minutes if we have more questions.
Okay, if there's no more questions, thanks so much to Gabrielle for those really great insights and sharing your own research as well. Really enjoyed that part.